So the goal of this presentation is to introduce a recent article that we published in Trends in Neuroscience entitled Asymmetry between pre- and postsynaptic transient nanodomain shape neuronal communications, written with my partner Martin Heine. So the goal of this presentation is to introduce what we think can define nanodomains in neuroscience. So again, other presentation will be available on my YouTube channels, and some of the notes are in bionumetry.org. So basically summarized in this review, we were interested in what defines, using single particle trajectory, what can define a region where receptors, but not only, like calcium ions, are restricted. So I will decompose this lecture into several points. The first one is neuronal nanodomain definition using calcium ion channels and receptors. Then we will go to the empirical definition using single particle trajectories. Then, to define nanodomains, I'll explain the concept of potential wells. And then we will see an applications to the pre- and postsynaptic terminals. Then we will move on to discuss Nano Column as an example of nanodomain organization between two cells. Then we will discuss how glial cells can create diffusions inside specific nanodomains. And finally, we will ask the question is, can this specific nanodomain, can they be defined as phase separation or phase separated aggregates, as a terminology coming from genetics for understanding how the nucleus is organized. And finally, we'll discuss what are the role of these nanodomains between the molecular and the subcellular scale. And the idea is that they can be defined, their role is to put order into molecular disorder in order to perform cellular function. So, point one, what are these neuronal nanodomains? How can they be defined? So, to discuss this, I would like to take the example in here of the synapses between a pre and a postsynaptic neuron, a post and a pre. And what if we look specifically at the region in the presynaptic terminal where we have a magnification here? And what is interesting is that when calcium flows inside the channels, calcium seems to be restricted in a specific region here underneath the vesicle and the plasma membrane here. And the consequence of this is that the calcium can specifically bind to specific binding site in important molecules such as synaptotagmin that will influence or trigger the release of the vesicle by interacting with a snare complex. So how come calcium ion be specifically restricted, or their concentration increases in this specific region? So it is known that if there are binding site or multiple binding site here, and calcium can bind to this binding site, then if you look at the density, so this is another calcium ion, if you look at the density, we know that multiple binding site can create a region where the concentration here is going to be, so this is the concentration of calcium, is going to be increased. So multiple binding sites can create this effect, but also the fact that there are restriction barriers by the plasma membrane. So this is an example of how a domain such as a calcium nanodomain can exist because of the particular architecture and the binding site of this domain. Another example is if we consider the case of mitochondria, where we have here a very beautiful chiome reconstruction. And if we look specifically at the interaction between the mitochondria and the ER, the endoplasmic reticulum here, and we look more specifically again at this interaction of these two membranes, this MAM, we have here a region where calcium can be sequestered a sufficiently amount of time so that when calcium is released from the ER here and move into this region, the calcium can flow inside the mitochondria, which can regulate the production of ATP. So again, the interesting part is by having this restriction of the region of the order of tenths of nanometer, we have a specific nanodomain region. So now we are going to converge to the main focus of this review, which is if we look at the electron microscopy of synapses, we have here region, this dense region, suggesting that there are here accumulation of specific numbers of molecule protein channels that have a particular function in the definition of the synaptic current. And so when explored by single particle trajectories, these trajectories, so a molecule can be labeled, a receptor can be labeled, and move the motion on here that I'm showing now, the motion on the surface can be monitored. And if we look at the different type of motion, we see it's from one very confined to four not confined at all, from motion that can oscillate between the two. But what is interesting is that not all parts of a domain, of a nanodomain, so this represents here the postsynaptic density, the surface of the postsynaptic density. But what has been found here is that not every part of the postsynaptic density is explored by trajectory, but only a small part of it. So you have here a static picture where you see the receptors, again, located in this nano region. And the most intriguing question is, how come this receptor can be at the same time restricted, but on the other hand, they can be exchanged? That is, some that are outside here are going to move inside, and some that are inside are going to move outside. So this is another example coming from calcium voltage channel trajectory. So here you can see I'm going to change color now so that you can see the region in purple, which corresponds to many trajectories, and you have on the top of this circle that corresponds to region where trajectory are accumulated. And to see this, let me go to the figure on the right here, where we have shown as the color code. So the color code is like, when it is red, it's like 10 particles per few tenths of nanometer. We can see region of accumulation of the receptors. And the question is, what defines this nanodomains region that are observed by collecting all of this trajectory and put them all together on the same graph. So this seems to be true across many membrane proteins. So this is an example of GAG here, but we have seen the calcium voltage channels, where in all different cases, or also AMPA receptor or even calreticulin inside the the ER or the neuromuscular junction, we see a region of high density, that is, particles, receptors are not uniformly organized, but they are going to be aggregated in some specific region where they can actually be exchanged. So what is the underlying mechanism of this retention of these receptors? So we had defined across the last 10 years different scenarios. One scenario that leads to the narrow escape problem is to say that the confinement is basically due to membrane. Membrane is enough to confine transiently a particle that you can see here moving by Brownian motion until the particle escapes through a small door. And using the classical formula and associated diffusion coefficient, the time, this can lead to a time of about a few seconds. Here it's about a minute to escape. And if obstacles are positioned in between the entrance and the rest of the domain, the time can increase. But one thing is for sure is that if we need to increase the number of receptors, for sure, this mechanism is not enough. We need specific binding, a change in the number of binders inside. And this has been explored a couple of years ago theoretically. Another possibility to retain receptor is by thinking about using the classical potential well structure, which is well known in chemistry, where a potential well is a force that restricts the motion of a particle. So a particle enters into this, it falls into the receptor, and then it's very hard for the particle to escape. So it spends most of the time here at the bottom, and by chance it can escape here. This is what the potential looks like in dimension 2, and this is a signature in when we look at the gradient of the field. So can we extract this structure from single particle trajectories? So the answer is actually yes. From many single particle and redundant trajectory that are passing through the same point, here's the magnification, and this is an example of several types of calcium voltage channels. It is indeed possible to extract these vector fields here, as you see, and even more, we can extract the boundary, and the boundary is quite important to define the energy that has been reconstructed here. So we see different potential wells with different energy. So when a receptor enter into this region, they are going to be trapped for some times. I insist that this is only possible if we have a redundant number of trajectories. This is what is needed for the statistics to work and for the reconstruction to give reliable numbers. So in conclusion, we saw that the fact that there are converging arrow shows that the potential well is the main structure for retaining this the receptor. However, the exact mechanism remains open. So what are the role of these nanodomains? So to address this question, in a recent article published in Neuron, my partner Martin Heine and his group showed that actually these receptors were colocalized, where you can see in this figure here, you can see neurons, and the white part corresponds to a specific marker of the synapses. And what was interesting is that the region where we have high concentration of this trajectory, we saw the response here, that you can see the calcium response at those regions that are marked here in these different nanodomains 1, 2, 3, 4, and 5, where you see that they are exactly colocalized where region where we see this high concentration of receptor. So this is an example that a nanodomain is not just an accumulation of a receptor, but this is a region where, in addition, this accumulation leads to a signal, like calcium signal, that is so important for mediating parts of the beginning of neurotransmission at synapses. And this is an example where we can reconstruct it. We have reconstructed the channels, the channels potential wells. So another interesting aspect is the fact that these potential wells are stable in time. And how we do this, you can look at it using a time-lapse analysis, where basically what you have is you have a time window, and the time window is shifted here every 10 seconds. So you can get trajectories that were not taken in the first window. And by doing this across time, you can see how stable, this is the red ellipse here. You can see how the red ellipse is stable and can disappear. And by looking at the region where we have a continuous, across a different window, a continuous presence of the ellipse, we can mark the time where we detected the potential wells. And when we look at it for a different calcium voltage channel, we found that the time scale was of the order of 30 seconds, confirming that these nanodomains are stable in time. So nanodomains not only are stable in time, but as you can see here, they can deform, they can change locally their shape. So finally, to define these nanodomains, we can use the size, the energy, the diffusion coefficient. However, if we looked if there are any correlation between size of energy, of diffusion coefficients of energy, this ensemble of points states that there are no specific correlation between these different parameters. So finally, what is quite interesting is to compare the type of nanodomain that can be extracted at the presynaptic terminal, such as the one we extracted from calcium voltage channels, or other, such as the one that can be extracted from a parareceptor and NMDA receptors, any kind of postsynaptic receptor in excitatory or inhibitory synapses. And what is interesting is that there are major differences between those two types of potential wells. In the presynaptic terminal, the size is of the order of 80 nanometers. The diffusion coefficient that we could find inside is 0.05 micrometer square per second. The time of stability is about 30 seconds. And the residence time of the receptors is hundreds of milliseconds, but it's still much longer than the time for free diffusion. And we think that this guarantees a fast redistribution following vesicular fusion. In comparison of the pre with the post, the postsynaptic potential wells are much bigger, hundreds, 200, 400 nanometer compared to 80. The diffusion coefficient is about the same, but the stability of the well is much bigger, of the order of tens of minutes compared to half a minute for the presynaptic terminal. The residence times is of the order of seconds, much longer, and the energy of the well associated is larger than the one in the presynaptic terminal. And we think that these guarantee a much longer stability of the receptor. Basically, in the presynaptic terminal, due to vesicular fusion, you need a fast redistribution, which is not needed here in the postsynaptic terminal. Probably this region will be more important for long-term changes. So another important aspect of the connection between pre- and postsynaptic terminal came from the theoretical studies about the fact that there could be a sort of alignment where you have a presynaptic terminal, you have here many receptors moving, but after some possible changes in the plasticity, what has been proposed is that there is an alignment where vesicles are going to be released and where the post-synaptic receptors are going to be located. The first study on this was numerical, published in PNAS in 1997. And the paper mentioned the novel expression mechanism of synaptic potentiation with an alignment of presynaptic release and postsynaptic receptor. And in 2011, we made one more step in this, saying that maybe there is not only one, but there are several of these column. We mentioned at that time that from analysis, we cannot postulate that the synapse should contain multiple structures where vesicular fusion spots are opposed to one of several clusters of unpaired receptor. This was in order to guarantee an efficient transmission and having basically several pieces of several synapses in one. And interestingly, this theoretical hypothesis that came from simulation, but also here in the data of the analysis of miniature event, a couple of years later, in the group of Blampied, this was observed experimentally, where you see the alignment here of pre- and postsynaptic elements, unpaired receptor, and where vesicles are going to dock. So now another interesting part of nanodomains is how calcium can be restricted in this region, and how these nanodomains could, for example, be perturbed. So it is known that under some specific condition that glial cells can penetrate, or on the contrary, is prevented to penetrate into the synaptic cleft. And under some specific condition, when it did penetrate, it's so as we can see here in this connexin 30 knockout, where you see here the penetration of the glial protrusion inside the cleft here. So this could, for example, affect the nano column. When you have this penetration, the nano column here could be disrupted, and this could disrupt this specific communication between these pre and post nanodomains. So we have summarized in the review, I'm not going to spend much time on this, the different elements that characterize these nanodomains. Where are they located? What is the diffusion coefficient? The diameter of the nanodomains, the energy, the mean time of a molecule inside, which is a fundamental characterization of the stability. And again, this stability is the result of the statistical analysis and cannot be just obtained by a single trajectory. And then the summary of the stability. So we looked at this for voltage calcium channels, sodium channels, and receptors. The data are less clear for NMDA and calls for really looking at them carefully, and glycine receptors. Finally, I would like to compare these nanodomains with what is known in the literature as phase separation, which is very well known in the case of the nucleus. So this is an example of two types of molecules. Here you have blue, and here you have the yellow one. And under some condition, the blue aggregates into droplets. And these droplets is called phase separation. So here you have this example. And basically the phase separation is a phase which excludes basically one type of molecules and have all of one on another type located in the same one. So this is here shown in this example. And this phase separation are quite stable. So this is a cartoon of what can happen with chromatin. You can have chromatin binder that are going to bind chromatin and create specific loop. This is again obtained by bridging factor, where you can have loops, and by creating this loop and also this self-associating chromatin binder, we can have part of the chromatin which is very condensed, expelling all kind of molecule, of small or large molecules, and possibly this has a genetic and this could carry genetic information by preventing some specific gene to be activated. And so one question came a couple of years ago, is that are the nanodomains we have studied in neurons, are they the same as the one that are sought in the case of the nucleus. And we think that this is not the case for the following reason, that if we think about this nanodomain that can retain here receptors, they need to be transient. That is, receptors should go inside and outside. We don't understand how this is regulated, but one thing is for sure: the energy here could be regulated, and this is what we saw from single particle trajectories. The shape can change, and these nanodomains must be open in the sense that they should continue regulate the exchange in a very specific manner of the receptor in order to make sure that synaptic transmission is possible with the right amount of receptor in order to have the current that needs to be generated. And this is an example for during a plasticity protocol. If you want to have more receptor, and this is like LTP, long-term potentiation mechanism that could lead to more receptors, so increasing the probability of release in the presynaptic terminal. This would be like LTD, where we could imagine that the energy of the well is diminished so that the time the receptor spends is shorter. In the postsynaptic part, this has already been shown that LTP, LTD is associated with a regulation of the number of receptors. What defined exactly those numbers remains unclear, but the potential well energy plays a fundamental role on this, as it defined the residence time of the receptor inside these nanodomains. So from here to here, we would go with a type of LTD protocol, which leads to a change in the reduction of the size here, size reduction, and thus the number of receptor inside, or the residence time is lower. The receptors spend less time here. What defined the number of receptor here, we thought in the past, has to do not only with the potential well, but also with specific anchored protein that could also participate to the genesis of this potential well. This mechanism remains unclear to the moment. So finally, what are the role of these nanodomains? We said that if we have many molecules, many receptors that are moving with Brownian motion, so it costs basically no energy to move them. But the price to pay to put order into synapse is to have this structure. That is, if they move by Brownian motion, in order to find them at the right place, you need a structure, and it costs energy to build such a structure. And the role of this structure is to put order into the synapse, to have receptor at the one place. They could be controlled, the number could be controlled, the residence time is controlled, but also the size and the energy of this potential well, as well as the precise location, as we have seen with these nanodomains, could contribute fundamentally to their position. And with all of this, the neuron should be able to perform its function and have the specific physiological response that define its function. On this, I would like to finish this presentation, and thank you for listening.